to come in at the last moment because we had a space in the schedule, but I thought it was a great opportunity to have Alex along because Alex is, I think it's official now, Alex, is it not? That he is joining us at the University of Manchester as a chair in AI this month. I don't know if you've actually started yet, Alex. Probably like in these last few days. And Alex has been doing some really fascinating work on in silico regulatory science and in silico trials, and really sort of merging ideas in machine learning, digital twins, modeling, and regulation of clinical trials. And I think he's got a great story to tell about this. So without further ado, over to Alex.
Super. Thanks a lot, Magnus. And yes, I'm in Argentina at the moment. We are running a conference here in the Andes the next week, and I was visiting a bit family in the week before. So what I'm going to do today is there is limited time, so I had to trade between breadth and depth. And what I try to do is give a talk that hopefully will finish earlier than the time we have, so that there is an opportunity for you to ask questions, and I may be able to go in a bit more detail in those areas that you may have interest. But what I want to achieve today is to try to give you a new vocabulary for a few topics that maybe you're not familiar with, but also at least give you a glimpse about how that connects with machine learning and AI, and therefore how a lot of the things that I'm sure you've been hearing and discussing this week actually have a massive impact into areas that are quite important. So first, I'd like to talk a bit what we do in the area of computational precision, computational medicine, which is the name of my chair, and I'm going to be putting some names of people who are very close collaborators over a number of years in developing some of these concepts. So one thing that applies to most of the work that we all do in machine learning, whether it is in silico trials or regulatory science or not, is this pyramid, which is actually a pyramid from the 1960s from information management, and that shows that there is a pyramid of abstraction in the data, going from data, which is the information we get from sensors, we can digitalize into information where we actually give contents and structure it, to knowledge, which is given meaning to that information and synthesize, provide learning. And then ultimately, this is all amassing information from the past, data that we have from things that the most is current from the minute that you do the measurement. But most of the times what we want to do in the area of healthcare is make future decisions. And that requires to have something that we call wisdom, that is validated knowledge. So if you think about in medicine, data could be the temperature or the blood glucose levels. The information could be, the fact that that patient could be diabetes. The knowledge is that diabetic patients in a particular trial have tended to have worse prognosis, and wisdom is the fact that you have now clinical guidelines where you have validated the knowledge over a number of studies, and upon those, clinicians can really take decisions in terms of future of the patients or management. And that's something that is very important, because we put a lot of information sometimes, a lot of emphasis sometimes to data, but actually decisions is what clinicians really care about. And and there are bottom-up approaches, where you go from data towards decisions. There is mechanistic approaches where you understand, what is happening with the physics or the biology or the physiology, and you try to make sense of individual measurements. And that is what gives sense to a number of AI techniques that we are familiar with, and I'll come back to them in a minute. So if you want to read a bit more about some of these ideas, there's a couple of papers that we wrote where we established, the definitions about what precision imaging is, and fundamentally around be it more descriptive, predictive, and integrative, and also the differences between simulation and synthesis in imaging. Now, we talked about phenomenological and mechanistic models, and basically it's all about data versus knowledge. is all about sampling, which is what limits your ability to collect data, versus the theory, which is kind of what limits you in your knowledge for developed mechanistic models. And there's a number of concepts that to me are actually quite important. One is that as we go over the next few years, and this comes from a model developed by Der Helbing in ETH, as we go over the next few years, the amount of computational power we know that follows the Moore's law and doubles every 18 months roughly. The data is actually currently doubling every 12 months, also in exponential form. And people talk that it won't be long before it actually doubles every 12 hours. But it still remains being an exponential one. And the important thing is that when you have two exponentials of different coefficients, one is faster, there is at some point a crossing point, which is here, where the data you will have will be much bigger than any computational power you will be able to collect. But what really is the killer is that if we look at what we call systemic complexity, and this could be the complexity of the biological or health systems that we study, but also the complexity of the networks and models that we build in terms of number of parameters, they follow a factorial growth, which is much faster than any exponential you can come up with. And what essentially this means that there will be a point, not too far again in the future, where the amount of complexity we are trying to understand is much bigger than the data we can collect, let alone the computational power we have. And this is a fundamental Achilles heel of any data-driven approaches. We just simply can't just depend on data-driven approaches. Many people believe we can. I don't believe we can. Now, the only way to deal with this is what is called the course of the scale is actually by incorporating priors, because priors allows you to overcome these links. It actually tells you that there are not that many free parameters. It's only important to collect as much data, and that perhaps you can do some computational tricks to accelerate things. And this is what we talk about knowledge-based or knowledge-informed deep learning, where you can trade off knowledge and mechanisms and availability of data in ways that you don't need to work on the sort of conventional machine learnings or pure mechanistic models, but you can combine it with both. And I'm not going to talk a lot about this today, but this is part of the work that we are doing as well that relates to how we're going to accelerate some of the things that I'm going to show you. So now let's move a bit into regulatory science and how we can make it more precise as well. So first of all, why we bother about this? Well, there's a study that shows that in a period of 10 years, there was on the order of 83,000 deaths and over 1.7 million serious injuries due to medical implants that have gone through the whole regulatory pathway. So these are not devices that haven't been tested. These are devices that got FDA, EMA, MHRA clearance. And despite of that, there is serious issues in harming those. And in the context of the UK, is what is called the Cumberledge report that actually has shown some of the limitations that MHRA has in actually picking up some of these challenging cases. Now, it's not just the problem, and it's not that it's a small problem, but it's not the problem only in terms of the people that we put at risk, but it's also that this is going to happen more and more because technology is becoming complex and there's a lot of technological convergence of combinations of AI with implants, with chemistry, for instance, in new devices. And this system we have for regulatory approval is just unfit. The burden just keeps growing. That is in one aspect while we have these serious adverse effects. And every time one of those happens, we have a serious impact on industry. It is told that about 10% of the shares of a company, the shares drop by 10% the day after recall of a new device is made. And that actually makes companies more vulnerable, R&D costs longer because they need to be more thorough, go through serious checks with regulators, and therefore the time to market keeps growing. And this becomes unsustainable because ultimately we are paying for that cost through our insurances, through our different national systems. So how can we solve this? And this is basically that we need a mechanism that can shorten this current life cycle that is from years. A device is probably 7 to 11 years. A drug is between 11 and 15 years to actually come to market, and with a huge attrition in the innovations. So what we want is an approach where we can fail fast and fail virtual and fail earlier, and with that address a number of other problems. Like, many trials are going out from the UK, from the UK to the US, because there is a better regulatory framework. The cost of the trials is enormous, so it takes about 6 to 7 years to generate evidence, and between 10 and 150 million. And what we want is a way that we can actually reduce those costs and bring those trials back to the different geographies, because otherwise we end up having devices that have been approved in a particular country, but where then the translation of that evidence to different countries is far from obvious. So just to give you a sense of the seriousness of this is a plot that probably you don't see much the numbers, but I just want you to focus on the trends. These are about 20 years worth of time. The first curve here corresponds to medical implants or medical devices. The second one is to pharma, and this is how much the demand for drugs and devices has grown over the years. And it's about 6 to 5, what is called compound annual growth rate. To give you a reference, automotive industry is about 5%. Okay, and then if you look at the same period of time and you look at how many devices and drugs have been approved by FDA in the US, you see that it's flat. So we have more demand, flat, output in terms of new technologies coming in. And this is a stifling innovation and a stifling, also a lot of good innovations that could make a difference to patients. To give you, also a sense of impact, a few months ago, AVHI developed together with CPI a report, 1 in 5 UK health techs, and 90% of the companies in the health tech domain in the UK are SMEs. So 1 in 5 of those are going to the US to do their product launch, because it's complex to do this in the UK. So what this all had to do with in silico medicine and computational medicine and in silico trials? Well, computational medicine is essentially applying methods from engineering, math, and computer science to improve our understanding of disease and their treatment. And in silico trials, and I'm going to focus on my second definition, is basically any computer-based tests or trials that use mechanistic prediction models on highly controlled virtual conditions and virtual cohorts that represents realistic operational conditions. So is this new? Well, in a sense, not a lot. I mean, if you look at the aerospace industry, we are doing a lot of virtual modeling and simulation, and this is a very complex, multi-scale system, highly regulated, life-threatening potential. We don't hear every day that a plane is dropping down, and there are thousands of flights every day. So, they're doing something good there. Automotive industry, another example. We don't tend to do, 30 years worth of innovation and led us to, Tesla and similar cars, but we don't do any more wind tunnels. We don't do so much, we do aerodynamic simulations. We don't do virtual prototyping. So we don't do physical crash tests, of prototypes against a wall. We actually do virtual prototyping. We do virtual crash tests, and we are talking about self-driving cars. And a few years ago, what I was wondering is why we cannot do the same thing in medicine. Why, if I show you 30 years worth of innovation in a particular device, and we're going to talk a bit later, which is stents, coils, and flow diversion devices for brain aneurysms, why 30 years worth of innovation is still a bunch of wires? Why we still have 20% complication rate and nearly 3% mortality rate with this technique? Why can't we develop simulations where we test the hemodynamics and the impact of that hemodynamics in different types of conditions? So same device, what happens if I extend it one way, the other way, or with both ways? And you can see that the magnitudes of flow velocity are actually heavily affected by what we do, and we haven't even changed the device. So, one thing we shouldn't do in something smart like this in medicine as well. And this is part of something bigger that is called, is a paradigm shift in engineering, which is called upfront simulation, where the idea basically is, this is adapted to healthcare, but on the x-axis you have all the life cycle of a product, and in the vertical axis you have the number of problems that you need to solve to go to market. So what we do today is what happens in the black dotted line, this one here. We tend to do very little testing early on. So a company very quickly wants to freeze their design so that they can do testing, because at this stage, they don't even know whether they have a product. So any test they do is essentially a waste of money. They need to have some preclinical trial and clinical trial to have that evidence. Then they do more testing. The problem is that if something needs to be fixed here, they need to go back to the drawing board, and that is more expensive. That's why in this phase, the cost per problem fix is much bigger than at the beginning. And then they go to human trials, and then here the costs are massive for many reasons, including potential legal claims that they need to address. So they do very little testing at the beginning, a lot of testing at the end, where it's very expensive. So what we are predicating is this purple line, where we try to do testing early on in a virtual way, so it's very cheap, we can do it at scale. What we want to do is testing under conditions that are as close as possible to the one we will find in human populations, and that's where the AI and large data analytics will come, that I'll show in a moment. So that by the time we go to the clinical testing, we have everything that we could have identified early on, we have identified. There is always going to be things that we cannot identify. We only focus on those, and hopefully the costs are lower at that stage. So let's try to show this a bit more in practice. So imagine we have a device like a stent, like we talked before, and aneurysms, and imagine that I get access to a very large scale database. This could be a regional database from imaging. So regions like Manchester, like Leeds, have catching bases in their hospitals of 3 to 5 million people. And we're doing imaging from all of those. So imagine that if we do a query of all the images in that database over the, that have anything to do with brain vasculature over the last 10 years, you get something on the order of 20,000 subjects. We've done these experiments in Leeds. And now for each of those, imagine that we throw some algorithms from image segmentation where we can really start generating models, detailed models of these different aneurysms and the feeding vessels. Okay, so I'm just keeping here a small portion of the whole vasculature. And then what we can do is for each of these guys that we have here in black, we can generate a 3D model. Again, you need to do AI and machine learning to do this analysis, and then produce virtual implantations of these stents. And we can put the same stent in 2 or 2,000 different anatomies, and then look at whatever the index of quality is. In our case, what we look is at the reduction in blood flow post-implantation in relation to the blood flow that was before the implantation, right? Because that gives you a measure about how much you have reduced the blood flow, and as a consequence, how much clotting you have produced within the aneurysm, which is what we want to do. Now, you could do this. I'm trying to get this back. So you can do this, and then you can say, well, now we want to also look at yeah. So we also want to look now. We also imagine now that we have wearable sensors from pages, and maybe some of you work on signal processing and machine learning for signal processing. So you can develop models like ocean process models, like those ones where we, so here we had about 100 subjects that we measured them with holders over 48 hours. And what we are building here is the mean blood, the mean velocity magnitude from Holter measurements, the variability across the 100 subjects we had. And then we actually, because we had 48 hours, we could create models at different time points. And what I'm showing you here is the average value at rest and the average value at a stress, and the variability over stress and over rest. Okay, so you can take each of these anatomical models and put the same device, but now what we can put is different regimes of physiology, so the rest conditions, exercise conditions. And you can imagine this might not be even ethically possible in a real population, because these people may be elderly, that you can just put them into a machine to run or do a pharmacological stress test for ethical reasons. And you can see that the quality metrics is again different depending where you are. And then finally, of course, you can bring different devices, and you can do the same. You can have now the same geometry, the same physiology, but now different devices are looking at the effect of different devices between themselves. Okay? Now, what you have here, if I put it all together, is like a massive cube of experiments, of virtual experiments, where in one direction you are modifying the anatomy, in another direction you're modifying the physiology, or you're modifying the device. And I'm being very generic here because each of these axes has multiple parameters that you could modify. But hopefully you get the idea that each of these nodes becomes a particular anatomy and a particular physiology with a particular device. And if we would color code each of these nodes with success or failure in terms of the response we anticipate, you actually have a map of performance of the device across the population. And you can do an inverse readout of this matrix and have a stratification map that tells you which patients would be best responders of which devices. Okay? So how do you generate this? Well, from large databases that are increasingly more and more available. Like in my case so far, I've been working with UK Biobank, with Yorkshire Imaging Study, which is the 3 million people PACS systems, list care records that provides you a lot of physiological information and so on and so forth. So fundamentally, an in silico trial has 3 main ingredients. Has a way to generate virtual participants and populations, and this is very connected with things like generative models, synthetic data, and so on. Ways to build virtual examinations and virtual interventions. So examinations if we are doing in silico trials of imaging, or interventions if it's in devices like I showed before. And then you also need to have virtual readers and virtual outcomes to have an inkling about how that particular placement of the device will lead to a good or not so good response. So what we see is that in the future, rather than having the current situation where most is bench, animal, and human testing, by increasing the amount of virtual populations and computational models, we might be able to reduce the amount of animal, bench, and human testing and focus it there where it makes the biggest difference. We are not advocating the full removal of those. What we are suggesting is sometimes you might be able to, to reduce the number of tests to those that you are comfortable, that are most likely to be good responders. That's what we talk about: replace, reduce, and refine conventional trials. So in the future, what we believe is that regulatory evidence, so we talked about regulations, or I talked a bit about regulations. What we mean in the end is relatively simple. By that, regulatory science is the science of providing better scientific evidence for regulatory approval. But what fundamentally people look into regulatory approval in healthcare is that the devices are safe and they are efficacious, right? So you want to know that there is no side effect potentially from that device, and also that if you put that device is better than whatever you are doing in the as best a standard of practice today. And what we believe is that in the future, these will be a blend of evidence, partly coming from models, partly coming from bench and animal and human testing, but where we should only go to human and animal and bench testing where essential. So maybe one comment here to make is that let's not forget that animal, bench, and human testing are models. They're not the gold standard. They're models of performance and efficacy and safety of devices and drugs, but they also have all kinds of issues. Animal testing not always clearly translates to humans. Human testing, puts humans at risk. Bench testing is very expensive and is very different to in vivo conditions. Okay, so what we are suggesting is that we have to have a fourth option that actually gives richness to this scenario. Okay, so to give you a few examples now about case studies, this vision now into action, and in particular to show this that was nicely put in this paper, as simulated trials provide depth and nuance to randomized controlled trials gold standards. So randomized controlled trials is one form of clinical trials that is understood to be the closest to a gold standard you can think of. So what we will see is that in silico trials provide additional depth and nuance to those results. So I'm going to skip a bit this one because I'm going to go in a lot more detail later on, but this is a trial that we did last year on flow diversion devices in aneurysms. But there is also people who are looking, that's why I talk about trials and testing. There are people who are developing models for replacing bench testing. So if you are in the orthopedics space and you want to check the safety of a device and the performance, before you go to humans, you go to cadaveric specimens where you insert the implant and you put them into a machine that does bending or does a stress testing. And that in itself is very expensive. It could be easily, each of these tests, £100,000. So can you do that also with modeling and simulation? The answer is yes. And there is work by industry with Zimmer Biomet, for instance, which is an orthopedics company. The PUI, which is another orthopedics company, working together with colleagues in Bath, developed also another in silico trial for a surgical plus device combination. So this is what is called osteotomy, which is they cut a piece of the bone and remove like a wedge so that they can make people sort of to walk more straight when they have a problem, malformation from birth, and they need to remove a part of the bone, but they also need to stabilize with a certain device, as you can see here, a certain implant. And these colleagues actually use these sort of techniques to then do sizing and generate some early evidence before they went to human trials. And then even FDA, as regulators, are developing in silico trials of imaging, where, for instance, they build models of the breast and in mammography, and then they have very good models of the physics formation from both tomosynthesis and conventional X-ray, and they are able to generate different sort of breast anatomies and through the physics, implant, look at the images that are generated, implant potential tumors that are obtained from real subjects and then sort of synthesized but integrated within the synthetic images as well, and eventually use this to look at the ability for humans to do readings in these images and what modality gives you the most important evidence. Okay, so this is just to give an example that before I go into my own example, that you realize there's a lot more people working in these across different devices. So the device where I've been working most for over the years has been on aneurysms, brain aneurysms. Brain aneurysms are a bulge in the vasculature that if it bursts, you generate an hemorrhagic stroke. And what people do is they put different type of devices inside, coils or stents or potentially clipping, which is like a clip you put in the neck, to effectively remove from the circulation the aneurysm, either by robotic coagulation selectively within the aneurysm in the minimally invasive approaches, or in the case of clipping, which requires craniotomy, so you need to open the skull for this one, to remove it fully by putting a clip. Well, this is more or less the same, but just that you see, they like to appear mostly on the circle of Willis and in the bifurcations, and these are the different techniques. Maybe the one I didn't mention is sometimes you can also do a stent with coils, where you use the stents in wide neck aneurysms to keep the coils in place. Now, there are tons of these devices, so when I tell you about the stents, we're going to be talking about one of them, but imagine that there may be easily 20 of them being developed at the same time. And if we could figure out through modeling that half of these will never make the cut, they will never really be successful, this is that many trials that we avoided. and we focus on the potential winners. So what is the mechanism of action of these devices? So these flow diverters are very tightly packed struts that divert the flow, and by diverting the flow, what they do is they stagnate the blood within the aneurysm and produce selective clotting, which effectively removes the aneurysm from the circulation. There are some small vessels called perforators or side branches that one needs to be very careful not to close, because those feed the brain, and if you close them, you produce an ischemic stroke as well. I'll mention this because we'll come back in a second to that. Now, to give you an idea of how this all worked, this paper in radiology was what is called the pivotal trial, so the trial that allowed this company to go to market, that took 8 years from design to publication. So imagine you have the whole device ready to go, but you need to get these pivotal trials. You need to spend 8 years, 6 years recruiting patients, because you need to follow up them for, say, 5 years. And this is only to acquire 108 patients, and probably is burned between 20 and 40 million pounds in that period, most of which goes into the follow-up of patients and research nurses and so on. They claim this device is one of the most studied. This is a device from Medtronic. We did this work unrelated to Medtronic, but POOFs, which is this trial that I mentioned before, is 106. Premier, this was for small aneurysms, for large aneurysms. Then they developed Premier, which is another 141, that was for large aneurysms. So we have already the behavior in the small, we just need to check whether for large aneurysms it would work. And then Aspire, which we'll talk a bit as well, this is a registry. So something you do once you go to market to keep an eye on what's going on with the subjects. Okay, 100 dice. So if you put it all together, there is about 500 subjects. Okay? And for instance, our hospital in Leeds used to do 100 of these patients a year. Okay? Imagine there are tons of those hospitals in the world. So what we did is we developed a pipeline. This is kind of the sort of AI pipelines that we need to use, where you go from the images to the segmentation, then you need to do proper cleanup, running, volumetric meshes, do the deployment of the devices, set boundary conditions for the blood flow, so what are the inlets and outlets, all those curves that I was mentioning at the beginning. Then you need to solve Navier-Stokes equations into a number crunch machine or using physically informed networks if you can. And then you have the physics, so the blood flow, and then you solve the biochemistry, which is basically all these coupled to the flow PDEs. And that gives you an idea about the quality of the plots. So if you do that, and you need to trust me, just because we don't have lots of time now, but we didn't have access to the data from these trials. So the only thing we could do is take another data set and demonstrate that the demographics was similar to the one of the original trials, which was the case. And this is basically what we show here in this plot that I'm going to skip. So what was the main conclusion? The main conclusion is, sorry, one second. Okay, can you hear me now? Okay, sorry, I need to sneeze. So the conclusion was that with this approach, we could reproduce to a 5% accuracy the result of that human trial. The human trial took 6 to 9 years, I mentioned. We could do this in 3 months, okay? Once you have all the pipeline in place, running it actually could be faster because it's how much cloud access you want to pay for. And then we could demonstrate not only that we could reproduce those results, but that we could look at the data. So these are the results. FD passes our in silico trial. We predicted an efficacy of 75% overall efficacy, while the other trials are more or less around the same numbers. If you look at the gray part here, it says NT and HT 83 and 67. So NT is normal tensive, HT is hypertensive. So because we have control of the physiology, we could simulate what happens when the 88 subjects are all hypertensive, all of them are normotensive. This is not possible if you have databases which are as small as the ones from these trials, because they didn't even correct it for blood pressure. So what they have is an average performance, but what we could show is that actually if you have normotensive patients, the performance is on average much better, it's 83%. But if you have hypertensive patients, that performance goes down, and therefore the proportions you have of normotensive and hypertensive will bias a lot the results of your study, and also mask the effect of hypertension. So without going into the details, because if you're not into the aneurysm space, probably that won't mean much to you, but what this plot shows is that for a number of questions that in the literature have been posed by a number of researchers, but where there is no trial to demonstrate, like what happens when you have a branch artery? What happens when there are large or giant aneurysms as opposed to small and normal aneurysms? What happens when you have aneurysms with a very high aspect ratio? So what we could actually demonstrate is that we could do subgroup analysis and start to elucidate the behavior in those scenarios. So an in silico trial is not just a replacement of a conventional trial. Sometimes it can give you information that goes beyond. One of those, perhaps the most exciting one in my view, is the fact that we demonstrated that when aneurysms have a side branch, remember the side branch from the beginning? Look at case number 3 and case number 4. These two subjects are the same subject. They have the same anatomy. The difference is that this guy is a normotensive phenotype, and this is the hypertensive phenotype. And we show that in 27, which are all the cases we had with side branch of the 88 cases, in every case that there was a side branch, and in every case the side branch subject was hypertensive, what was happening is that the clot was starting to form also in the side branch as well. And as a consequence, you would clot that branch and you would produce an ischemic stroke. While in all of the subjects where there was a side branch and there was we put a normotensive phenotype, there was never the side branch being clot. Okay? So that's quite important, because you can start thinking about combination therapies where you give anti-hypertensive to subjects ahead of doing the intervention for a certain period of time. You control the pressure, and then you put these devices. Okay, just to give you a second example on a bit faster, but this is coils. So these are coils. So coils are made of a number of materials. Nitinol is one of those. And some of these materials, you can give them a shape memory. So you can pre-shape them, then you put into the guide wire as a straight, and when you take them out of the guide wire, they recover the original shape. So they can start filling the volume of the aneurysm, okay? Or you can have them as sort of a spring type, and then you sort of need to sort of whip them into the aneurysm. Some of them also have hydrogels around that are prothrombotic, but we're not going to focus on that today. Now, you may wonder how on earth we can predict where these guys will end up, because this looks like very stochastic, very chaotic. Well, that's precisely what we do, is let's assume they were a random field, right? Where there's only certain things we can control. So what we develop is a method that essentially allows to insert all the coils within the aneurysm with some rules and some properties that we learn from real data. And then we fill the aneurysms with those rules, and then each color of these coils corresponds to a different coil, because they have a certain length and diameter, and you know how much of that you can fill into the aneurysm, okay? So I'm not going to give you a lot of explanation on the validation with it, but I did that slide to show that there's a lot of validation behind this. So we validated our algorithms against histology, and we validated our algorithms against time dilution curves in real angiograms. So what I'm going to show you is the value of all of this in the end. So imagine each of these columns in this plot, you see below it says 20, 25, and 32%. So what is that? This is a parameter that is used in the clinics, which is the packing rate, is the volume of the amount of coil over the volume of the aneurysm itself. Okay, so it tells you how much metal you're putting. And here you're putting 20%, 25, 32. You see that already with 20, it looks like fairly packed, and actually it's almost impossible to go about 33%. Each of the colors corresponds to one different thread. Normally you can put into an aneurysm, depending on the size, between 6 and 30, and it's about 1,000 pounds each of them. So putting a few more or a few less is a bit of money as well. And what we have done is, so that is each of the columns. Each of the rows says configuration A, configuration B, configuration C. So this, if you think in terms of random field, each row is a realization of that random field. Okay, so this is effectively, if you look at column 1, configuration A, we put metal to 20% according to our algorithm. Then we take it out, which we couldn't do in a real trial. Then we put it again in configuration B to 20%, but now it will fall into a slightly different position. We take them out and we put it back in configuration C, and you can do that as many times as you want. Okay, so if you do that, and you do that with more density of packing rate than I show here, what you have is plots like this that show you the average flow reduction. So of course, as you put the metal, you slow down the flow as a function of the packing rate. And what you see is that when you are in very low packing rates regimes, all of the different realizations have very different values of the flow reduction. But as you go into increasing packing rate, that starts to stabilize into growth. Another group with humans demonstrated that aneurysms that have that are stable on the long term, so that they don't reoccur, they have an embolization that is on the order of 32 to 20%, while the same guys, well, this depends on if the aneurysm has a small neck or a wide neck, they're large, they're small. So in all of the same cases, for unstable aneurysms, the packing rate is smaller. And this is understandable, because if you put a packing rate which is very low, you are very much operator dependent, and your outcome will be very depending on exactly who you did it and how much you really felt and so on. If you put too many, there's two possibilities. You put really too many, in which case you probably burst the aneurysm and you will not count that guy, or you got closer to the maximum that is beyond which there is no dependency on the blood flow, and then you basically have a good ceiling of the aneurysm, which is the higher values that they recount here. So this thing, even in a chaotic environment like CODS, what allows you to do is to find the optimal patient-specific threshold beyond which it's not worth to put more coils. You are spending money on potentially risk of bursting the aneurysm, and below which you don't want to put, because you know that you may have a reoccurrence down the line. Okay, so in this case, what we are testing is not the device, but the procedure as a whole. Is the device may be the same, but depending on how you put, you will get better outcome or worse outcome. Okay, so I'm conscious about the time that we have now left, so I'm gonna leave 10 minutes to answer questions, but I just want to show a couple of the slides. Only to say thanks to the team. We have a large team that is working in various elements of this, and that I presented the work of some of them. We're also looking into how we can generate virtual populations with synthetic models, where we can put, also comorbidities or covariates to do the equivalent to patient selection, and also to be looking into ways to accelerate all of those simulations, because the minute we need to start doing thousands and millions of those, then you may not want to do them through conventional finite elements, which is what we do at the moment. And I think I'll stop here for questions.
Okay, Alex, can you hear me okay?
I can hear you.
Okay, so, I'm not sure if Alex will be able to hear the question. Okay, so Alex, I've got a question actually. It's a kind of general question about the sort of how important modern approaches to machine learning have been in realizing this in silico trial vision. So machine learning has really kind of accelerated in the last decade, I think. So maybe I wonder if you could say a little bit about how much these different steps in constructing these in silico trials have been influenced by the most kind of modern developments in machine learning?
Yeah, I think that's a very good one. And also helps me to go through some of the slides that I haven't shown, where perhaps some of those points were a bit more clear. I think there are two main areas where AI and machine learning are crucial. One is in the scaling up of these trials. So I show you this first in silico trial we did with 88 subjects. I think that was a relatively small in silico trial because you know, with all the story I told you, you would probably have liked to see on the order of 8,000 subjects, virtual subjects, right? So I think where in silico trials are going to be particularly exciting is if we can show that we can have, say, 2 to 3 orders of magnitude larger trials than we will have with any conventional trial. So the minute you want to do that is where scaling up becomes essential for in silico trials. And I'm showing here a slide where building these digital twins with the old tools that we had, say, 10 years ago was very, very time consuming. So we use most of those tools actually to do that trial that I showed. And that means that you needed 6 weeks to actually do 82 cases analysis. This is from running segmentation, mesh generation, and mainly the issue is that you need to double check that all of those steps are correct. And conventional tools are prone to a lot of errors. And by conventional tools, I mean conventional segmentation, conventional meshing. Now we have a much better pipeline that we are developing for the new trials, where we can do about 30 minutes per case and is fully automated. So we can do hundreds of those, okay? And just to give you an idea of what one of those pipelines looks like, it looks like something like this. Say that you already did the segmentation, which is a pre-step. You still need to detect where the aneurysm is automatically. You need to compute center lines. You need to find where the neck of the aneurysm is. You need to deploy the aneurysm. You need to do volumetric meshes. And you need to also set boundary conditions for which you need to know the label of the inlets and outlets of these models. And you need to do that on hundreds of those models without trying to do anything manual of any type. And that is really, really challenging. So we develop algorithms that are actually based on deep learning networks. And then, we always do a lot of validation, which I'll skip now. We also develop techniques that actually are able to segment the algorithm from the whole segmentation, because that's where we want to then compute, average flows for the metrics of success. We also, I talked about putting boundary conditions. Well, the boundary conditions depends whether the aneurysm is in this location or in this location or in this location. They will be different. So what we have is a wireframe type of model, which is a graph, and then we need to fit any geometry from any subject to that graph. But to do that, you need to realize that circles of Willis have 8 topological variants, which are all of them actually normal subjects. Some of us don't have certain terminals. So essentially it's a 1-to-8 or a 1-to-many graph matching, where sometimes you only get partial information because most of this imaging comes from rotational angiography, for which you only have one side, as you normally inject the contrast from one of the jugular veins or the other ones, of the carotid arteries, and then as a consequence, you only see half of the brain in each of those. So these are algorithms where colors here mean labels of positions, and we automatically segment those using these graph matching techniques, where we use well, all kind of differential properties like angles and distance and torsion as clarification. And then the other thing is that if we only rely on data that comes from trials, we'll always be limited to the inclusion and exclusion constraints of those trials. So one of the things we've been doing over the last few years is developing techniques that we can use population studies like UK Biobank to build very large models of the brain on 40,000 subjects. Now, the problem with UK Biobank and with many other population studies is that they don't like to use any invasive technique for the imaging because all of the people are volunteers normally. So you will have nice images of the brain, but not angiographic, so you don't see the vessels. So we develop techniques where we are able to actually produce, I think this is probably a better one, where you are able to see the vessels from a non-angiographic image. So one of these two images, this is a Turing test for you guys. One of these images is the real angiographic scan. The other one is the angiographic scan that we synthesize on the same patient based on the non-contrasted images of those. And I can tell you that it's difficult to figure out the difference between them. So we've done techniques that actually are you could see them as similar to the ones about the style transfer type of techniques, where we learn the mappings between angiographic and non-angiographic, and we apply that to very large populations of non-angiographic studies. So anyway, hopefully that gives you a bit of an idea, backgrounds of all of those are different ways of using machine learning and deep learning.
Alex, which one is it? You've got to tell us the answer to the Turing test.
So this is the synthetic one, okay? And when if I would, well, maybe you guys, you see it in a very large screen, but you need to be really, you can see it in very details like these structures you have here, for instance. We are now doing a study with radiologists where we're going to be synthesizing the two, the modalities, and then ask radiologists to detect them automatically and see what is the kappa statistic for those. So it's quite interesting. I always say that for me, developing the algorithms is important. It's usually about a third of the time we spend. But then developing the validation is another third, and developing the understanding about what systems we need to develop is the other third of the efforts, where our energy goes.
Great, thanks. Any questions for Alice? Yeah.
Hi, Alejandro. Thanks for the talk. I was wondering how far are we from using the same techniques, but for more complex organs like the heart or lungs?
I didn't hear the organs. I think you're asking whether we could use similar techniques for other organs, but I didn't hear the ones you.
It was heart and lungs. Heart and lungs.
Oh, heart and lungs. Yes. So actually, I recently got awarded an ERC Advanced Grant for working on TAVI, which is transaortic valve implantation, and I'm not showing them to you, but we do have already synthetic models of the heart that we built over the UK Biobank on very large cases. And we also have developed in those ways where we can say synthesize only females from 45 to 55, or from 45 to 50 years old that are smokers. So, but by combining synthesis not purely on the image alone, but the image plus all the metadata through joint latent spaces that we synthesize jointly. Lungs, we haven't done work in that space, but in principle is possible as well. One of the things that's why I didn't show you why in aneurysms we are working purely on databases where we have instances of real subjects, is that if you want to do a synthesis of the anatomy in organs that are arborescent, like in the vasculature of the brain, or the lungs will be the same, you need to come up with some smarter techniques for synthesis, and we are having some work going on in using diffusion models to do that, because you have topological variations into those organs. So to me, the division line is not whether the organ looks more complicated or looks simpler, or if it has a lot of deformation or not, but is that a fixed topology like the heart is most of the times, or is it a varying topology like the vessels or airways or guts could be? But there are techniques to do that as well.
Thanks. Any more questions? Oh, okay, well, thanks Alex once more.
Super. And I wish I could be with all of you guys. Make the most of it. I think it's a great event that you have. And for those of you in Manchester, hopefully we'll have a chance to talk soon. Okay?
Thanks, Alex. And get back to your family now. Okay.
Super. Thanks a lot.